Hello, this is Ogan Gurel, and this is lecture number 1 for Bioelectromagnetics, Overview and History. Bioelectromagnetics. We'll first start actually with Isaac Newton. Most of you are aware that Isaac Newton was the founder of much of modern physics with his book Principia, leading to the 3 laws of physics and the various other discoveries and theories that have laid the groundwork for physics. And if you look at this quote here, he talks about light and the vibrations of this spirit mutually propagated along the solid filaments of the nerves, from the outward origins of sense to the brain, and from the brain into the muscles. These are the final words, actually, of his Principia. And at the very end, he says, but these things cannot be explained in few words, nor are furnished with that sufficiency of experiments which is required to an accurate determination and demonstration of the laws which this electric and elastic spirit operates. And that's the end of the Principia. The point here is that Isaac Newton was actually quite fascinated with biology. And although he's rightly regarded as a major founder of modern physics, his interests were very much in biology. And this course actually is neither about biology alone nor physics alone, but a combination of these, or the intersection of physics and biology. And in particular, to understand a little bit more how this electric and elastic spirit operates. So what is bioelectromagnetics? Bioelectromagnetics, also known as bioelectromagnetism, is the study of the interactions between electromagnetic fields and biological entities. Areas of study include electrical or electromagnetic fields produced by living cells, tissues, and organisms, the nerve impulses, bioluminescence, the cell membrane potential, electric currents. Others include animal navigation that uses the geomagnetic field, and of course, the effects of man-made sources of electromagnetism, from mobile phones to X-rays to lasers and so forth. So it is the intersection or interaction between electromagnetic fields and biology. So bioelectromagnetics is not a new field. In many ways, it's a frontier, but there is an official journal of the Bioelectromagnetic Society and the European Bioelectromagnetics Association. And as they quote, they specialize in reporting original data on biological effects and applications of electromagnetic fields that range in frequency from basically 0 hertz, cycles per second, namely static fields, to the terahertz undulations of visible light. And of course, there is X-rays and gamma rays, which we'll also talk about in this course. So the Bioelectromagnetic Society promotes the exchange of ideas to advance the science of natural and applied bioelectromagnetic fields in biology and medicine. So this is fairly well established. So before going into the content of the course, let's talk a little bit about the mechanics. We'll discuss some methods, requirements and grading, the readings, the course content, and the schedule. And we've split apart the course in 2 parts, part 1 and part 2. So what are the core methods? We'll have lectures as well as a special topics discussion. We may have some possible guest lectures. The lectures offline, namely in person, are scheduled for Friday afternoons, 2 to 4 p.m. But during this situation with the COVID-19 coronavirus, we'll be doing these lectures online. Lecture notes will be posted on the web in the Google Calendar, as I've informed you via Kakao Talk and email. We'll talk about that in a little more detail shortly. The attendance is per digit regulations, and all lectures and discussion will be in English, and participation is very important. So let's now talk about requirements and grading. Attendance accounts for 20%, and we'll figure out methods to track that. whether it's listening to this lecture, participating in some online Kakao Talk group, or submitting weekly short papers. We'll talk about the attendance. Discussion and participation. We'll have a very short write-up prior to each lecture. And at the end of this lecture, I will describe the write-up that is to be prepared for this, before this Friday. And the final paper, which will be 30% of the grade, will be to analyze and critique an original scientific paper in the bioelectromagnetics field. The format will be determined, but it's roughly 5 pages, not a very long paper. And we'll discuss that later in the course. So as far as readings, the core textbook is a comprehensive set of lectures, and you can see that in the Google Calendar. So here would be lecture 1 in your Google Calendar, and you can basically just click on the PDF and the lectures will be available in that form. We will also share, either by YouTube or some other platform, these online lectures. Supplementary reading may also be assigned, and here in your syllabus, and you can find the syllabus in every lecture on the Google Calendar. This is the syllabus, this would be the lecture. You could find some other books. This one is free, but this is not required reading, it's supplementary reading, and there may be some additional papers to be assigned. I believe I have all of your Gmail addresses, preferably Gmail, because that will be easier to integrate with the Google Calendar and other resources. So you can contact me. This is my number. This is my email. My office is in E7201. It's unclear when we will have face-to-face meetings, but in general, my office hours will be Friday, 4 to 5 p.m. The content in the course. As mentioned, bioelectromagnetics studies the interaction between electromagnetism and biology, namely how electromagnetic phenomena are involved in biological processes, both biological processes producing, generating electromagnetic fields and waves, as well as how external electromagnetic fields interact with biological molecules, cells, tissues, and organisms. This is an extraordinarily diverse subject, requiring strong knowledge in both physics and biology, and spans a wide range of phenomena such as the generation, propagation of nerve signals, the molecular events underlying photoreception, how we see, bioluminescence, cancer radiation therapy, the electrocardiogram, EKG, magnetic resonance imaging, and much more. And this course will provide an overview of this diverse field, with the first half covering the basic principles, which will include today's overview in history, some core concepts in electromagnetism and biology later in the lectures, as well as a review of key electromagnetic processes in biology. The second half of the course covers a wide range of applications of bioelectromagnetism, building on what we discussed in the first half, including the fundamentals of the interaction of radiation with biological organisms on the non-ionizing and ionizing side, electromagnetic methods of diagnosis, EKG, EEG, EMG, ENG, etc., biophotonics, and biomagnetism. The course will conclude with more futuristic topics in bioelectronics, including protein electrodynamics, terahertz medicine, and biocomputing. So as mentioned, the course has 2 parts. Part 1, first half, approximately, will be the key principles, which will include today's overview, basic concepts in electromagnetism, basic concepts in biology, and electromagnetic processes in biology, going from the small, namely molecules, to cells and tissues, to whole organisms. Part 2 of the course will be the applications, and these include the interaction of radiation with biological organisms, general principles, the interaction of non-ionizing radiation in particular, and the interaction of ionizing radiation. And these two are quite distinct, and we will talk about the implications of that throughout this course. Then we'll zoom in on more specific topics, including electromagnetic methods of diagnosis, biophotonics, biomagnetism, bioelectronics, and the discussion of the paper's special topics. As mentioned, everything will be on a Google Calendar. So that's all been set up from today's lecture on February 28th, next week's March 6th, and every Friday and so forth. So you click on each of these to get any additional information specific to that date, including the PDF lecture notes, and it will implement the link to this video lecture. So in terms of principles, let's talk about some basic concepts in electromagnetism, some basic concepts in biology, and some fundamental electromagnetic processes in biology of molecular organisms. This is, of course, going to be a very quick overview, which we'll develop these topics in more detail with subsequent lectures. So basic concepts in electromagnetism. One key concept, of course, is the concept of electricity. And electricity, as you know, involves charges. And these charges have three important properties. These include the duality of charges. You're well familiar with positive and negative charges. And like charges will repel, have a higher energy, and opposite charges will attract, have a lower energy. The second principle is quantization of charge. Every charge is some multiple, for our purposes, of one elementary charge, which is about 1 times 10 to the minus 19th coulombs. And an electron in an atom will constitute one negative elementary charge, and a proton will constitute one positive elementary charge. Another very important principle, the third one, is the conservation of charge, which namely states that the algebraic sum of all positive and negative charges in an isolated system remains constant. This diagram indicates that charge can actually be created. So conservation of charge does not necessarily mean that charge is constant, but rather that the total sum of charges remains a constant. So in this box here, you have no charge. Some light comes in, and that creates a photoelectric effect that creates an electron, say, and a positron, oppositely charged. So you can see the charges are created inside this box, the isolated system, as it were, but the net charge is going to be constant. So duality of charge, quantization of charge, and conservation of charge. Second very important principle in electricity is Coulomb's law. Coulomb's law is the law that, in classical sense, underlies electric force between these charges as described. And Coulomb's law is analogous to gravitation. Because charges can be both positive and negative as opposed to mass, which is just a positive quantity for the purpose of gravitation, Coulombic forces can be attractive between unlike charges, and they can be repulsive between like charges. And this is the force law in different units. And you have a direction vector, a magnitude, and the product of the two charges. And very importantly, you have this inverse square dependence, which we'll talk about. So as I mentioned, inverse square dependence means that the force, as you get away from a charge, will go down with the square of the distance. And then also it's very important to note that for our purposes, charges are additive. You have superposition of forces. So if you have many charges, 20 charges, 30 charges, you just add up all the different pairwise interactions, and you get the net force by just adding all of these for the individual charge-charge interactions. So the second concept in electromagnetism, and again, all of this is very basic review, which we'll go into more detail in subsequent lectures, is the concept of magnetic fields. So 2 points are very important. First, magnetic field lines are closed, and second, that the movement of charges is key. In other words, magnetic fields and forces result from moving charges, so there's a close relationship between electricity and magnetism. Electricity or charges, when they move, more precisely when they accelerate, create magnetic fields and forces. And the converse is also true. Magnetic fields and forces can only act on moving or accelerating charges. So this would be the magnetic field due to a current wire, and the current is expressed by the letter I here. And this current represents moving charges as described. And then the magnetic field will be a closed loop at some varying magnitude given by this equation in a distance r from this current wire. In other words, the magnetic field will get smaller and smaller as you get away from the wire. Not an inverse square dependence, but in this case, just an inverse relationship. So let's talk about the force due to a magnetic field. So again, magnetic field is described by this B. It acts on moving charges. So it can act on a point charge, and the force is going to be the charge times the velocity. Remember, moving charges times the magnetic field. Or it can be on a current, and in this case, the current times the length that's acted on times the magnetic field. Again, don't worry about these details for now. The key point is that moving charges are related to the generation and effect of magnetic fields. Magnetic induction, or Lenz's law, is the concept that a changing magnetic flux, or the magnetic field through a certain region, in this case A is the region, causes a voltage or electromotive force. So it will cause a electric field. The resulting electromotive force creates a current, which produces a magnetic field that opposes the change in that magnetic flux. And this is given by Faraday's law. Phi B over T is the change in flux over time, and the voltage will be the opposite of that. So for example, if an external field is increasing into the page, as shown by this X, and this is a current loop, then the induced field will be generated coming out of the current loop. The third concept in electromagnetism, we talked about electricity, we talked about magnetism. The third concept is that of waves, and that brings both of these together. If you imagine this concept that of that Faraday's law, and again, think of it very conceptually, that a changing magnetic field will create a current, which will then create an opposite field. There's a close relationship between these moving charges and the generation of a magnetic field, and then the induction of a magnetic field, and so forth. So to put it very conceptually, the acceleration of charges will result in a changing magnetic field. A changing magnetic field, in turn, will result in a changing electric field. And the result, essentially, is a wave. So these charges are accelerating, creates a changing magnetic field, creates a changing electric field, and this self-propagating interaction, if you will create a wave. And this electromagnetic wave has, like any wave, a relationship between the wavelength and the frequency. That relationship is inverse in this regard. So for example, if you have radio waves, which have a very small frequency but a very long wavelength, so the frequency and the wavelength are inverse, and you have the highest energy gamma rays, which have a very high frequency, very high energy, and a very low or small wavelength. So again, wavelength is inverse to the frequency, and the energy is proportional to the frequency. So more frequency, higher frequency, greater energy, and this is called Planck's equation, as many of you may be aware. So if we focus on this electromagnetism onto the visible spectrum, the visible light is a component of the electromagnetic spectrum. So the electromagnetic spectrum goes all the way from radio waves to gamma rays. Visible light is in the middle here. Violet is the highest frequency, it's close to ultraviolet, while red is the lowest frequency, close to infrared. So again, violet radiation or violet light close to ultraviolet has a high frequency, a short wavelength, and a high energy, relatively speaking. Again, dictated by this inverse equation. And red light has a low frequency and a long wavelength, again dictated by this equation. So we've talked about electricity, we've talked about magnetism, we've talked about waves. Now let's talk about how electromagnetic waves or energy interact with matter. So if we brought this in very broad terms, we have what are called matter-matter and matter-energy-matter interactions. So when it comes to matter, we have and energy, we have three different types of particles. And this is a little bit of particle physics. We don't need to know the details now. We have bosons, hadrons, and fermions. The bosons are these photons, largely, that mediate electromagnetic waves. The hadrons include the protons and the neutrons, and the fermions include the leptons, which are the electrons and the neutrinos. So very light particles, heavy particles, and photons. So from the left side, we have energy, and on the right side, we have matter. So there's a kind of conversion between energy and matter. Most of you are familiar with Einstein's equation, E equals mc squared. And we have matter-matter interactions, which involve chemical bonding, for example. So a atom here will combine with another atom. The electrons will interact and create a chemical bond. And we can also have photon matter interactions. For example, photoionization. Energy comes in and an electron is given off. So this would be energy matter and matter-matter interactions. Our main focus will be on these energy matter interactions. In other words, how does this energy come in? In other words, more these bosons, in particular photons, come in and interact with matter, namely hadrons and fermions, and mostly these electrons, as we'll see. So photons interacting largely with electrons. Again, this is at a very conceptual level, but we will develop these concepts in more detail. So that's a little bit of physics. We reviewed electricity, magnetism, waves, and how energy and matter interact in the context of matter-matter and matter-energy interactions. So now let's talk about a little bit of biology. So the fundamental unit of biology is the cell, and in particular the eukaryotic cell, which is what animals and plants are comprised of, have all these components. Let's go through them very quickly. It looks like a lot of stuff here, but let's go through it systematically. One of the most distinguishing characteristics of the eukaryotic cell is the plasma membrane. The plasma membrane is the outside of the cell, and it's responsible for the cell integrity. And across this cell membrane, we can have information transport and matter transport. In other words, what's inside and outside the cell are generally separated, but there is, of course, some transport between them. How is this membrane made? It's made on what's called a smooth endoplasmic reticulum, and this is responsible, this is an organelle inside the cell, responsible for membrane synthesis. Now, the genetic material of the cell, in particular the DNA, is within the nucleus. And eukaryotic cells are typically defined by this defined or circumscribed nucleus in which the DNA material resides. How is this DNA expressed into the proteins that are the machinery of the cell? Well, these are protein synthesis occurs on the ribosomes, where the DNA is transcribed to RNA, the RNA goes to the ribosomes, and the ribosomes produce proteins. There are effectively two types of ribosomes that are free in the cell or intracellular proteins, or ribosomes that are attached to the rough endoplasmic reticulum, membranes that are inside the cell that are responsible for membrane or extracellular protein synthesis. Some of these proteins, such as hormones, antibodies and so forth can be exported, and they are exported through a further specialization of this endoplasmic reticulum called the Golgi apparatus, and that's responsible for protein export. Now, what about proteins being imported or other molecules being imported or processed? And these are done in other specialized organelles called the lysosomes that break down and separate these molecules that are imported. And then how is the energy of the cell produced? These are produced in other specialized organelles called mitochondria. And in particular, they will produce ATP to power the cell, the ATP serving as a high energy intermediate. So another basic concept in biology is that these cells are not just randomly associated around the body, they are organized into certain systems. We have what's called the ectoderm and the endoderm that are embryologically derived. The ecto means outside, endo means inside. And then we have the mesoderm, which is the in-between. So in the ectoderm and endoderm, we have essentially epithelial type tissues and nervous system or neural type tissues. And the mesoderm, which is kind of the filling between the ectoderm and the endoderm, we have the connective tissues and the muscles. So all tissues in the body, all organs, can fall in these four categories. So we have a very complex organism, but in essence, we have epithelial and neural cells derived from ectoderm and endoderm, and connective tissues and muscles derived from mesoderm. Biology is organized in a hierarchy. So we have all the way from molecules. We'll talk about the bioelectromagnetic interaction of molecules. We have cells, as we talked about them. We have tissues and organs. Those tissues and organs are organized in terms of the organism, population, populations, and finally ecosystems. So now let's zoom in on this lower layer of molecules. Very important molecule, of course, in biology are proteins. Proteins are the machinery of cells. They are how we think, how we move, how we digest, how we produce energy, and so forth. And here are some representative proteins. These proteins, such as myoglobin, which is an oxygen storage protein, to basic fibroblast growth factor, which is a growth factor protein. They are organized in these ways. These are chains of amino acids, and this is mostly alpha helix. We'll talk about that in more detail. This is mostly beta strand. Here's another example of a van der Waals surface or atomic representation of human growth hormone, mostly alpha helix in this ribbon diagram, and the androgen receptor in this second example. Another very important molecule in biology, of course, are lipids. Lipids are what constitute this cell membrane. And lipids are composed of both a hydrophilic part, this yellow section here, and a hydrophobic region, the green region here. And hydrophilic means it can be readily dissolved in water. Hydrophobic means it avoids water. And so if you are in a liquid or aqueous environment, the hydrophilic and hydrophobic regions will organize spontaneously to maximize the connection to water or avoidance of water respectively, and they were organized into these membranes with the hydrophobic regions together as depicted here, and the hydrophilic regions separate as depicted here. Embedded in these membranes are various proteins, and these proteins are transmembrane proteins, integral proteins, and on the surface of the cell, they will often have carbohydrate or sugar side chains called glycoproteins. One particular example of molecules or proteins in this lipid are transport proteins. You can see here a voltage-dependent potassium channel. So this is the membrane. It's stripped away, and you can see that this channel is opening up to allow potassium to go through, and the protein is actually shifting to open up for the potassium. And the shifting of this protein will be dependent on the voltage. And that's why we call it a voltage-dependent potassium channel. Going back to now the next level, cells and tissues, we talked about the 4 tissue types: epithelial, neural, connective tissue, and muscle. When it comes to neural cells, there are 2 types of neural cells. There are neurons and there are glial cells. Neurons are the basic unit of structure and function for neural tissues. The most notable quality is irritability, the ability to produce an electrical signal in response to an external stimulus. And glial cells generally serve supporting functions. So this is the basic neuron. The neuron is composed of a cell body and a axon. On the cell body, impulses arrive as a kind of local potential, and then the impulses will be transmitted as a kind of action potential. And these local and action potentials are electrical signals. Now, to begin our story of how the electrical signals are created, we have to understand that there is a resting potential in the cell, all cells, including neurons. And this resting potential, this is the membrane, has a net slight increase of negative charges relative to positive charges on the inside. So this is a separation of charge. A separation of charge will, the net charge of the whole thing is 0, approximately so, but a separation of the charge in between them will create a voltage difference. This separation of charges occurs because the cell is actively pumping in potassium and actively pumping out sodium. So the inside of the cell has relatively high potassium, K+, and the outside of the cell has relatively high sodium. So all cells have this sodium-potassium gradient with a high potassium inside and a high sodium outside. Because the cell, remember we showed the potassium channel, but that's the voltage-dependent potassium channel. But because the cell has an intrinsic permeability to potassium, this excess potassium that's on the outside will travel to, on the inside, sorry, will travel to the outside. That's because of a chemical gradient. Anytime you separate some molecules by the laws of thermodynamics, they will try to spread themselves out evenly. Separation in that way is unnatural. It's kind of like if you're at a beach and you're digging a hole with sand, the sand will want to come back in. And that's, of course, a gravitational gradient. So anyway, the potassium goes from the inside to the outside, and because this is positive, it will create a net negative, slightly net negative on the inside. So it means that the resting potential is dominated by the electrochemical equilibrium of potassium, typically around minus 75 millivolts. So this is that resting potential as described, about minus 70. Now, when the cell has some signal at some certain threshold, it will suddenly shift to a positive side. We call this an action potential. So what is it that creates this positive on the inside? This is a depiction of that action potential that suddenly shifts from a negative in the resting case to a positive here. We will talk at a subsequent lecture in more details on how this happens, but basically the cell is like an electrical switch. It goes from a negative resting potential to a positive but transient electrical potential. We call that the action potential. When we go to organisms, we have bioluminescence, for example. And how does bioluminescence occur? You have a luciferin protein under ATP, and in this ATP reaction, it will drive the production of light. Oxygen, carbon dioxide is given off, and green to red light, yellow to green light. And we'll talk about how this occurs in more detail. So this is a protein. There's a molecule here which will have visible light characteristics that's connected to this protein, and you can have different types of light generated. We also have an example of biological batteries, and this is animal in the Amazon that generates very powerful electrical, the electric eel. So this is basically, imagine those action potentials that we described, or arresting potentials, negative and positive. You know, from batteries, if you put them in series, it will increase the voltage. So for example, one of these is -70, two of them will be 150 approximately. You have several of them, you have 300 millivolts. If you have hundreds of these, you can have thousands of volts generated. Again, batteries in series, you add up the voltage. Batteries in parallel, you take the, add up the inverse of the voltage. So here's the eel from the head to the tail. All these are stacked up, as you can see here. Electrolyte stack from tail to head increase the voltage, and multiple rows in parallel increase the current. So now we're going quickly into the second part of the course, into some applications, and we'll talk about the interaction of radiation with biological organisms, some electromagnetic methods of diagnosis, biophotonics, biomagnetism, and bioelectronics. And again, this is a very quick overview. So how does radiation interact with biological organisms? Well, we have medical effects, we have biological responses, we have physical chemical interactions, and physical interactions. So this is very important to consider. There's two basic categories. There are physical interactions. For example, you have ionization, you have excitation of particles, you have physical chemical interactions, formation of free radicals, DNA damage. So these are physical chemical interactions. And then you can have the biological interactions that relate not to the physics, but some sort of biological response that can be as a result of these physical chemical interactions, such as repair mechanisms, but there can be direct biological effects as well. So one example of an application is transcutaneous electrical nerve stimulation. It's a low-frequency electric current used to stimulate peripheral nerves in order to relieve pain. And so by altering the waveform, the frequency, the rate, the pulse width, duration, amplitude, intensity, etc., you create a stimulation of nerve fibers that, in a sense, confuse the nervous system to not respond as much to the pain nerve response, but to be distracted, as it were, by the electrical nerve stimulation. And again, we'll discuss this in more detail too. Another example is radio frequency ablation. Radio frequency is a radio wave, but if you do it intensely enough, you create heat. And if you have this needle that creates a high intensity radio frequency, it creates heat that will kill, for example, a cancer cell near that. You will use ultrasound, which is not electromagnetism, it's sound waves, to localize where that needle should be inserted. Now, those are examples of non-ionizing radiation. We also have ionizing radiation. And the ionizing radiation has deterministic effects. So in other words, at a high dose, short time, you'll get a certain direct effect. We call that deterministic. And then you have stochastic effects. The deterministic effects are direct physical damage. The stochastic effects are low doses. They occur over a long time. There's essentially no threshold, and you have some sort of DNA damage that occurs randomly. That's why we call it stochastic effects. Among the stochastic effects, we have indirect action, which can be via free radicals. So the ionizing radiation ionizes a water molecule, for example, to create an oxygen-free radical, and that causes damage, or direct action directly on the DNA molecules. So we can also use this radiation to kill cancer cells. It's a field known as radiation oncology. And this is a complex field that you don't just blast radiation in there. You have to balance 4 different elements. The body is repairing or responding to this radiation. Because most of this effect is that indirect or oxygenation free radical effect. As you damage the tissues, it's gradually being re-oxygenated with blood flow. The cells are not always in a sensitive state for being damaged by this DNA mutations. They are most sensitive in the actual DNA synthesis phase. So you have to balance with when the cell is in the cell cycle. And then, of course, the cells grow back, normal cells or cancer cells. And so you also have the dynamics of the cell population increasing. And that's why we have what's called a fractionated dose of radiation, a certain fraction of dose to minimize damage to normal cells, and fractionate over time as well to accommodate or account for these dynamics. That's fractionated dose. You can also imagine this deterministic effect, very high density of radiation at one time, and we call that radiosurgery. So you might have various radioactive sources with gamma rays, and they are distributed around so that their deterministic effect on normal tissue is minimal, but their deterministic effect on a focused target is very high, and this could be the tumor as described. We have various electromagnetic methods of diagnosis. So we talked about the electric action potential, and those action potentials occur in the heart. And it turns out there are 2 types of fibers in the heart. There are these specialized fibers that produce this action potential so that the working fibers or the muscle fibers can contract in an organized way. So that means there's electrical activity around the heart, and we'll talk about this in more detail in a later lecture. This is the action potential for the working fibers. You can see it's a little different than that action potential we saw for nerve cells, and that's because these charges are moving in and out in different ways. And again, the details will come later. Now, if we have this resting potential here, the inside is negative, and during the action potential for a certain period of time, the outside is negative or the inside is positive. That means if this is occurring over the heart, a certain region will be depolarized, a certain region will be resting, because the heart doesn't contract and electrically active all at the same time. I want to just go through this diagram one more time. The cardiac impulse, the electrical impulse, begins up here and goes over a certain period of time of hundreds of milliseconds through the rest of the heart. So there's a wave of electrical depolarization. What that means is that if you put various electrodes around the heart, you will see a difference or a dipole, and that dipole can be picked up by electrocardiogram. Another aspect of electromagnetic diagnosis is X-rays. So X-rays were discovered in 1895 by Röntgen, and those are high energy electromagnetic radiation that interacts in a physical basis with inner electrons or high energy electrons. You had other medical advances in imaging, cardiac catheterization, coronary angiogram, CT scan, and more sophisticated CT scans. Some examples from biophotonics. Biophotonics is using the visible or ultraviolet areas. For example, for endoscopy, you put a tube down the body to image the inside, simultaneous light delivery and image collection by fibers that are inserted into the esophagus or the colon or the urethra. Spectroscopic analysis, so you're looking not just at the visible image, but at specific frequencies, biosensing, light therapy, photodynamic therapy, laser microsurgery are examples. Here's photodynamic therapy for acne. And the basic idea is that you have a drug and it's activated by light that kills the bacteria that is in these acne glands. Magnetoreception, birds, as you know, are migrating and they go north and south, and they have specific magnetoreceptor cells in specialized organs, both birds and fishes. Another example of biomagnetism, of course, is MRI, and this is an MRI image, and the basic concept is that a patient is placed in a magnetic tunnel, radio waves are passed, so this is not very high energy, it's low energy radio waves. The radio waves will shift small energetic transitions in protons that are present in all the molecules, in particular spins of hydrogen protons, and these will be transformed into two-dimensional images based on how this energy that's input in the radio waves and the proton spins relax meaning they go back to their ground state. So you can distinguish between fat, bone, blood, air, tumors, cerebrospinal fluid, etc. And we'll talk about these in a subsequent lecture. Now, CT, as you know, is X-rays. So you can see here the X-rays are absorbed by this. The X-rays are absorbed by, are denoted by a very white image. This, of course, being the cranium or skull, so it's absorbed by bone. You also see a little bit more absorption out on the surface, and that's because of the cell bodies or soma of neurons are here, and they have a lot of mitochondria. So bone has calcium, which is a heavy atom, which is absorbed by X-rays, and the mitochondria have cytochromes, which have iron in them, which is also absorbed by these X-rays. With brain, this white and dark is not analogous, it's not due to heavy atoms, but of course to the characteristics of the protons. But you can see distinguished the white matter, which is the axons, and the gray matter, which is the cell bodies. We have biocomputing with applications in biocomputing, forensic, optical, electrochemical, semiconductor, signal transduction. We can make actually a kind of logic gate using these molecules, an AND gate, an OR gate, etc., which are basically the units of electronics. Finally, we have the concept of bioelectrodynamics, that proteins, those molecules we talked about earlier, emit and absorb radiation. Here is a picture of a protein, and you can see it's not static, it's actually vibrating. So proteins vibrate. We also know that the proteins are charged, and by what we talked about earlier, the Maxwell's equations, vibrating or moving charges will create electromagnetic fields and will also absorb electromagnetic radiation. So that means proteins are vibrating, they are emitting electromagnetic radiation and absorbing electromagnetic radiation. So let's finish up with one quote. There are more things in heaven and earth, Horatio, than are dreamt of in your philosophy. This is from Hamlet, Shakespeare's Hamlet. So many of you may be familiar that the most famous quote in Hamlet is, "To be or not to be, that is the question." This is actually my favorite quote. There are more things in heaven and earth, Horatio, than are dreamt of in your philosophy. I call this the Hamlet principle. And the concept is, if we look at something, that is our philosophy, that is our worldview. We must always consider that there's something beyond that. And so many of us, many of you, are physicists, are focused on physics. Many are focused on biology. And one of the concepts of this course is that our respective philosophies, there are more things in heaven and earth than are dreamt of in our own areas. And so the challenge and opportunity of this course will be to expand our horizons accordingly, following this Hamlet principle. So this week we talked about the overview and history, and next week we will go into more detail on the basic concepts of electromagnetism, the nature of light, quantized state of matter, light-matter interactions in spectroscopy, and definitions of these terms: scattering, absorption, diffraction, transmission, reflection, refraction. So the question for next week, so this is due by 2 p.m. on Friday. You should send me by email one basically paragraph with your name in Hangul and English, your picture, so I'll have some reference point, hopefully when we meet, and answer this question: how does light, in other words, electromagnetic radiation in general, not just visible light, how does electromagnetic radiation interact with matter? Just put it in your own words. Don't copy and paste from Wikipedia or anything like that, but just try to be, in a very succinct 2 or 3 sentences, your answer to this question. And please email that to me by 2 p.m. on Friday. That's it for today's lecture. Thank you very much. And please take care and stay safe.